100 participants. I don't want to make more than 100 people wait on the line. Thank you for being so incredibly on time. It's 1 minute past the hour, and let's make a start. So my name is Parker Moss. I'm the Chief Commercial Officer at Genomics England, and I'm one of the leaders in the cancer research practice. I'm so delighted to be hosting you today and hosting some of our really very interesting speakers who I'll introduce you to today. So this is, just to let everyone know, this session is being recorded, and we make all of these sessions available on our YouTube channel, which you can just find by searching for on YouTube for Genomics England. And all of our past talks are there. There are some really terrific ones. I do recommend that you peruse our back catalogue of terrific talks. So why are we doing this? We are trying to draw attention to the quality of the research that is done on whole genome sequencing data by the GSIP community, by the Genomics England Clinical Interpretation Partnerships, which are the academic groups that we form to study the data that we've aggregated as part of the 100,000 Genome Project, and that we're doing prospectively now in partnership with the NHS in the Genomic Medicine Service. And we also have private sector partners who have access to our data, who make huge contributions both on their own, but also in collaboration with our academic partnerships. And collaboration is something that we are really trying to drive and have been doing successfully in both cancer and rare disease. We have a real emphasis on patient engagement and the patient impact of the use of our data. So we would really encourage any researchers or any private sector biopharma partners who are interested in talking about their experience of working with our data or with genomic data in general, and who believe they have a message that would be instructive to the patient community and the research community, that they get in touch with you. We're always looking for interesting and provocative speakers. So do please let us know if you want to present at a seminar. We have some interesting seminars coming up. Zan, if you go to the next slide. Yeah, on the 15th of March, we're giving a presentation on how to use our current research environment. So for any of you who would like a bit of coaching on how the mechanics of our research environment work, please do come and join that. And then after that, we have, next slide, Zan. On the 29th of March, we have a really exciting presentation by Bill Newman on mitochondrial dysfunction. So we've already got a lot of interest in that talk. Please do put that in your diaries and be available to come and listen to Professor Bill Newman. I want to draw everyone's attention to the research summit that we are hosting on the 4th of May in the Design Centre in London. So we are absolutely delighted that this will be an in-person summit. All of our speakers are going to be there on the stage. We will have 4 tracks. We're anticipating about 1,000 visitors in person. We'll also be broadcasting for an international audience that want to dial in. And we're going to have a host of speakers talking about new technologies, about cancer research, about rare disease research, and about policy and ethics issues in genomics. It's going to be a great day, and it's going to be finally terrific to have an in-person audience so we can get together as a user community of our research and discuss the progress we've made since the lockdown in cancer, rare disease, and of course, COVID research as well. Okay, and so I think now I would like to introduce our first speaker, Dr. Vincent Plagnol. Vincent is a fascinating guy who works for one of our commercial partners, Genomics PLC. Vincent was educated in Paris, where he obtains his Bachelor of Maths at the École Normale Supérieure in theoretical probability. After his PhD in applied maths and computational biology at Southern California, he moved to the Juvenile Diabetes Research Foundation and the Wellcome Trust Diabetes and Information Lab at University in Cambridge to study genetics and autoimmune disease. I know he's also worked in private companies in early detection in lung cancer, and he joins us today from Genomics PLC, where they've been working extensively with our data. They have a real focus on polygenic risk scores, and I'm really excited to hear what Vincent has to say. So welcome to the stage, Vincent.
Thank you. So it's my cue. Let me share my screen. Hopefully it's readable for everyone. Okay, so thank you very much. So first of all, really happy to be here and excited to tell you a bit about what we do at Genomics PLC and how we work with Genomics England cohort. So my talk is about a tale of 2 UK cohorts development, validation, and application of polygenic risk cohort in GeL and UK Biobank. And those two cohorts have a lot in common and some differences as well. And I'll try to highlight how they work together. So first of all, an introduction to tell you why it matters and what we're trying to achieve. So first of all, what is a polygenic risk score? So a polygenic risk score is the accumulation of a large number of variants, typically common and typically with small effects. It may vary, some may be larger, some may be effects, some may be rarer. But in general, it's looking genome-wide. And so that's where whole genome sequencing come into play, of aggregating thousands of variants together, putting all of those effects and assigning each individual an aggregate score that is a sum of all of those effects that you've found all over the genome. So what you can do with that sort of information is stratify a population into various subgroups: individual at risk, individual at average risk, and individual at low risk. And you can see happening here on this graph on the top right corner, which I hope you can see my pointer. Let me know otherwise. Where you have, so what you see is, age of participants, that's UK Biobank data, and you see the percentage of the population diagnosed with type 2 diabetes in that case. And you can see that individuals at high genetic risk for type 2 diabetes have a lifetime risk in the range of 40%. The average individual in the UK has about 10% lifetime risk, and individuals who are protected for type 2 diabetes are around, maybe 2% or something like that. And today, in the absence of the knowledge of that genetics, individuals in the red curve or individuals on the blue curve are treated the same way. We see the same attention, the same prevention, and we think that's not optimum. We think we can do better and improve the care of those patients and the prevention and screening by allocating resources more efficiently. So I've told you about type 2 diabetes, but it happens for almost any disease traits or trait in general. Here are some curves from breast cancer on the left and prostate cancer on the right. You can see that there is a substantial amount of women who are at something like a 30% lifetime risk. We do believe that those women should deserve some more aggressive and earlier screening to pick up breast cancer earlier. And prostate cancer on the right, similar sort of picture for men, with some men at 40% lifetime risk and some at a much lower risk of something like 2%. And again, how can we leverage that information to do better prevention and screening in the NHS and beyond, across the world? How can we bring those insights to do better? One particularly interesting example, which I'm raising now, which is coronary artery disease, because we have a trial running on that, so more on that in a minute. You can see the effect be present both in men and women. There is much more coronary artery disease, heart attacks, essentially in men compared to women. You can see that the top range is 45% in men compared to 17, 18% in women. But broadly speaking, the separation of the curve is comparable across genders, and therefore there is potential to improve cardiovascular prevention in men and women. So, and what we're doing, to tell you a bit why it matters to patients and why it's important, we try to, at Genomics PLC, we really want to translate the scientific insight into patient benefits. So we have the first Prs Clinical Trials. We work with the NHS to evaluate the implementation of risk prediction by combining PRS with current clinical risk tool for cardiovascular disease in primary care. It's a disease called heart, started in November 2021, is still ongoing. And the NHS has stated its interest and motivation to bring that sort of technology into clinical practice. And we're doing it for heart, but we would like to do it for a lot of other diseases. We'd like to improve prevention for and screening for common cancers, diabetes, eye diseases. There's a lot of opportunities to leverage this tech. And so why Genomics England? So where does GeL come in? So much of our development and validation takes place in UK Biobank with, for many obvious reasons. UK Biobank is an extraordinary cohort, but it's also quite biased. There is a healthy bias. The enrollment is, so the rate of enrollment was fairly low. So the population you see has a lot of, specificities that may not be representative of the population as a whole. And people ask us a lot about that. Genomics England is another UK cohort, which is relevant to us, given our UK focus. It's Genomics PLC is a UK company. It works on the same population, but with different type of ascertainment. So for us, being able to replicate our findings, show that we are resilient to those biases. We're not affected by biases that are UK Biobank specific. In general, non-European ancestry individuals are limited. Those cohorts tend to be very European ancestry focused. And Genomics England has a higher representation. It's a smaller cohort on average, but I will show you the numbers. There is more on average non-European ancestry individual in GeL than UK Biobank. And that's helpful. That allows us to add to the numbers of non-European ancestry individuals. Furthermore, whole genome sequencing is the future. We know that. We work a lot with arrays today for cost reason and scalability, but we see WGS as where it's going. And the GeL cohort gives us a window into that future, and that's quite important. Largely, and that's maybe underrated, but that's quite important, more and more UK Biobank is in all sorts of meta-analysis and GWAS, left and right. And that may create overfitting issues. Obviously, we are as careful as can be to avoid those issues, but they may occur. So we work with, we work hard to avoid that. And GeL is a perfect cohort because most of those GWAS have not made it into general, into those big community meta-analyses that are shared by the community. So that's my introduction. Hopefully, I gave you a sense of why it matters. So here is an outline of the core of the talk. First, an intro on PRS in general, why they matter and how they compare to other risk factors, why should we care. I'll tell you something about rare and common variant to give you a sense about whole genome sequencing. And then I'll tell you about some results comparing UK Biobank to Genomics England and how we use Genomics England for PRS assessment. So first of all, let me tell you a bit more about how much PRS matters and how they compare to other risk factors, because I think it's an important context for this talk. So the key observation which I want you to be aware of is that for most cancers and many other traits, I will show you that in a minute, but especially for cancers, the two major risk factors are really age and genetics. So it's a graph that my team at Genomics PLC has put together. And you can see here that you have this breast cancer, you have a measure of predictive ability of a range of covariates for breast cancer. And you can see that, there isn't a lot happening when you compare all those traditional risk factors to the role of the polygenic risk score alone. And in fact, if you add all those other existing risk factors to the polygenic risk score, you don't go much further. Even family history doesn't boost the prediction very much, largely because the polygenic risk score captures a lot of this family history. So while family history is the strongest risk factor on its own, it still is dwarfed by the predictive ability of the PRS. So I think it's important to see that in the absence, it's a bit different for things like cardiovascular disease, where you have things like lipid and other biomarkers. But for many traits, really genetics and age is what's driving your risk to a large extent. So here's a graph which you may find interesting. We've been looking at a range of human diseases, and we're looking at the risk factor for those traits, and we've played the same game of trying to quantify the predictive ability of those risk factors. And we've compared it to the PRS. And the vast majority of cases, PRS dominates the picture. PRS is, in most traits, especially if you look at the earlier, the younger adult range of the spectrum, it is the biggest risk factor. You can see, let's look at breast cancer, for example, I was just telling you about. There's a huge gap. Family history is the strongest it is genetic, but not only genetic risk factor, but there is still a huge gap between what you can explain with or without the polygenic risk score. So it really matters, you really want to bring that PRS information into your predictive models. And the other thing I want to mention, which is important background on PRS predictive ability, it really varies substantially across traits. So here is, so we're working with UK Biobank and GeL to deliver a range of polygenic risk score for the community to work with and use that as an investigation tool. And here we've benchmarked a range of something like 20 diseases, and we look at the performance of our PRS. On the x-axis, you have a measure of predictive performance across ancestry group, and you can see that the gap between something like ovarian cancer, which has a very low predictive ability, is one of the at the bottom of the list, and prostate cancer, which is at the very top of the list, together with type 2 diabetes, is substantial. So your ability to predict will vary substantially depending on your disease area. So I think we hope that this sort of data, those graphs, those benchmarks, can help you understand how and if polygenic risk score can be really relevant to your disease area. Another note is the fact that there is a lot of differences across ethnic groups. I mentioned that earlier. It's a well-known limitation of polygenic risk score. So the colors here mark major ancestry subgroups. And you can see in blue that in general, the performance in European ancestry individuals is higher than in other subgroups, especially individuals of African ancestry. So that's a point that is important to be aware of. And we're trying to mitigate that. It's a big part of the work that my team is doing, but it's largely data-driven. And this is where, again, data like GeL help contribute a little bit to filling those gaps. So that's my few words to introduce you to this effect size and the importance of polygenic risk score for predictive modeling. Now I want to tell you something about rare variant, partly because GeL works with whole genome sequencing. So we have access to rare variants and WGS data. And let me tell you how it compares to the array work we've done in UK Biobank. So first of all, we've been doing a lot of what we call analytical validation to show that we can use WGS data to compute the polygenic risk score. It seems obvious, but the biases that come with array data and the biases that come with WGS data are quite substantial. I mean, there are different QC issues, different variants are typed with different qualities, and some biases happen in one feature and not the other. So we've been doing a lot of comparison, and here is a graph that's part of our analytical validation work, where we show the absolute risk score difference as a percentage between for two polygenic risk score, in that case, cardiovascular disease and type 2 diabetes. And the bulk, so again, there is variability. I mean, there is noise. We absolutely acknowledge that in the computation of polygenic risk score. But the bulk of the data is within something like 3 to 4% of the target. So in general, when you compare WGS to array data, WGS data cannot be used just as is. So we had to work quite a bit on the GeL cohort to make it work to our workflow. So we had to perform an imputation step to fill the gaps and make sure that all the variants we need were available to us to do the work we intended to do. So quite a bit of preparatory work to make that possible, but it's work that we could do. And when you do that, you find that the WGS data are perfectly acceptable for PRS computation. So now a message I want to tell you is that the work of predictive analytics is quite different from the work of genetic diagnosis. And let me highlight how that's the case with a disease like familial hypercholesterolemia. So FH, as most of you will know that, it's a genetically inherited disease caused by mutations in a trio of genes, LDLR, ApoB, and PCSK9. It's fairly common, 1 in 250 individuals, and I've grown up at UCL. I did quite a bit of work on FH, and I've learned, what I learned there is that the risk of cardiovascular disease in individuals with those FH mutations is extremely elevated. So here is a data that I've taken from a CDC flyer. It's from a study in 2018, but the numbers that are being quoted are something like a 22-fold increased risk of coronary heart disease if left untreated. So, FH, huge impact. When you find those rare mutations in those genes, a very large impact on disease risk. However, so what happens in UK Biobank when you look at prospective cohorts? Well, the picture is very different. So we've done the work of finding those FH mutations. We've worked with experts in the field to identify that set of FH mutations. And when you look at it and you look at the prospectively, instead of looking retrospectively, you find that for coronary heart disease, you find that the odds ratio associated with those FH mutations is only something in the range of 3-fold, nothing like the 22-fold, which I grew up learning about when I was working at UCL on FH. And I think that's quite a striking finding. And others, we're not the only one finding that. I mean, it's quite broadly. No one has found any evidence that the sort of massive odds ratio that we found that have been cited by the literature can be found in UK Biobank or prospective cohort in general. So what is going on here? Well, here is a comparison, and it turns out that if you look at cumulative hazard curve, the risk associated with those FH mutation is roughly comparable to being the top 9% of a polygenic risk score. So if I can bring your attention to this, you can see that the actual risk of CAD diagnosis is about 20% lifetime, which is again not a very extreme range of the PRS. And similarly, even when you look at BRCA mutation, you find that the lifetime risk for BRCA is, in that case, it's a BRCA2 mutation, is around 35–37%, which I'm less familiar with BRCA mutation than with FH, but I think it's in low range of our expectation. So I think it's quite important and striking when you work on WGS data to realize that the work of prospective modeling, predictive modeling, is not the same job as genetic diagnosis. The statistics work in a different way. And what's going on partly is treatment. I mean, FH can be diagnosed. You can find very high lipid levels and you can treat that, which explains why some of the risk is reduced. And that's a great thing. We want to find those FH mutations and we want to treat them. But obviously, the argument does not apply to BRCA1 or BRCA2 carriers. And I do think that in many studies, not all, I mean, there are perfectly well-conducted studies in that space, but many studies tend to overestimate the effect of those rare mutations because they look at families that are loaded for other risk factors, including polygenic risk score, which tend to lead to an overestimation of the risk of those rare mutations. So a lot of the work we do in GeL, in UK Biobank and other cohorts is to understand what those mutations actually mean looking forward, as opposed to trying to diagnose them looking retrospectively at the impact of those mutations. So I think that's important context for WGS. And now I'll tell you a little bit about our work in Genomics England and how it compares to the UK Biobank work. So first of all, what have we done in GeL? We haven't used the whole cohort, and I'll tell you why. We started with an aggregate release version 3, I think that's the correct term, with something like 80,000 individuals. We've removed rare disease problems because in our view, those individuals were specific with rare genetic disorders and not the ones we wanted to use to benchmark our polygenic risk score machinery. We were left with 48K individuals. We've then removed related individuals to limit any bias due to relatedness. There were a few sex mismatches, and we end up with 40,000 individuals for which we've computed our PRS on. It's remarkable that actually almost 10% of those 40K are from South Asian ancestry. This proportion is substantially higher than when we see in UK Biobank, and I think we're very grateful for that because that gives us some sense of performance in this ancestry subgroup. So again, not the full cohort. We went through an imputation of that whole cohort. It's computationally intensive, so it's another incentive to not go too heavy on the sample size. So we try to keep it reasonable. And we choose some reasonable filters that give us what we think is an appropriate evaluation set. And the main finding is, when we've done this work in GeL, we've compared the performance in the 100,000 Genome Project on the x-axis to another measure of performance, the same measure in the UK Biobank. And in general, there is a very high concordance between those two sets. It's not better, it's not worse, but it's really a result we were looking for and we were hoping to see, which confirms the fact that our genetic predictors are robust to the biases associated with UK Biobank, and we don't see anything vastly different or reduced in terms of predictive accuracy. There is no overfitting, there is no dark corner. We could confirm the validity. So it's essentially a validation step that we've been able to achieve using the Genomics England cohort and leveraging the ICD-10 code data provided by GeL, matching the phenotype definition across both cohorts. An interesting feature, which I think is worth being aware of, is that the performance of those polygenic risk score varied quite a bit between the recruitment arms. So you can see that the cancer arm and the rare disease arms had quite a bit of variability. I think it's not unexpected because cancer patients have treatments and different again other biases that are very hard to get the bottom of. But I think there are some features of GeL which are interesting in that regard that pick up those different populations. You can see as a performance, a look at ischemic stroke, for example, where we do better in the cancer arm. But if you look at type 2 diabetes, we do so much better in the rare disease arm than we do in the cancer arm. And again, I don't fully understand exactly what sort of processes lead to that, but I think it's interesting to see that we can detect those subpopulation of the GeL cohort. And one thing so why do we deliver all those PRS UK Biobank? So first of all, part of it is benchmarking for people to see what's working, what's not working. But it's to enable this sort of studies that here we've taken, PRS for ischemic stroke in UK Biobank, and we leverage the incredible richness of UK Biobank to correlate this polygenic risk score with up to 3,000 biomarkers available in UK Biobank. And so we are in the process of delivering those polygenic risk score for the scientific community to use. We want the community to take advantage of those scores and be able to do those investigations. And I think we could do similar things in GeL. We have not done so yet, but we're excited by the rich oncology data in GeL. So you can see here, it's a fascinating graph. You could spend quite a bit of time asking yourself why time watching television is correlated with your polygenic risk score for stroke, for example, or some things are obvious, some things are less obvious. It's a great hypothesis generation mechanism. And we are working with GeL to also make those polygenic risk score available to facilitate this sort of investigation. And again, I see oncology as a field where there is tons of data in GeL that might be interesting for hypothesis generation in the future. And just to finish, 2 slides to wrap it all up, just to compare those 2 cohorts and to tell you how they match with each other. So obviously, UK Biobank is larger, it's 500,000 individuals. GeL we've restricted for that PRS release. We only look at a subset, but we are generating our PRS for the whole set. We're working with Lucas on making that possible. There is substantially more diversity in percentage in GeL compared to what we see in UK Biobank. So the contribution of non-European ancestry is real, and that allows some validation in South Asian ancestry individual, which is much appreciated. GeL does not have today the same incredible variety of biomarkers that's available in UKB, but there is quite a bit in the oncology world, including tumor data, obviously, and that's something we want to work with. And obviously, WGS data is very important, and that's quite important. And that's my last slide. To summarize, UK Biobank and GeL have a lot in common, but also different biases and features. They complement each other well. GeL is smaller with more non-European ancestries. Take-home message is that WGS and array data are largely interchangeable for PRS computation, provided that we can apply a final imputation layer. WGS offers incredible opportunities for predictive analytics, but be careful. The analytics of predictive modelings are not the same as genetic diagnosis, so we have to be really cautious in the way we use those models. We're very pleased that the GeL cohort confirmed the robustness of the PRS model validated in UK Biobank. It was a big test for us, and that we think it's a step toward deployment. And we also see more to be done in that space by leveraging the rich oncology data in GeL. And I'll stop that. Thank you very much.
Thank you very much, Vincent, for such a clear presentation. And certainly our team at Genomics can really enjoy engaging with your bioinformaticians. We have quite a few questions in the chat, which I'll go through. But any other people that want to ask questions, please do use the Q&A tab, and I'll try to keep an eye on hands going up. And if I could just take the privilege of asking you the first question. So we're delighted that you're focusing so much on cancer, which is clearly one of the major indications that we focus on alongside rare disease. I think the applicability of polygenic risk scores in the clinic is very well understood and well appreciated clinically in common disease. I think there's been some debate amongst oncologists about the clinical applicability of polygenic risk scores in cancer, not so much that they're sufficiently risk discriminative, but more about the meaningful context of that against the absolute risk of cancer. And I just wanted to give you an opportunity to address the cancer community and explain how Genomics PLC thinks this will be applied in the clinic.
So most of our focus is on common cancers. So a lot of the criticism which I've seen, which I think are somewhat warranted on the rare cancer spectrum, and indeed knowing that your risk varies between 1% and 2% is not very meaningful in practice. I think in the world of breast cancer, for example, it's very hard for me to argue that we should not be using tailored risk. So if I can if I go a few slides back, so that's pretty extreme here. The polygenic risk score matching a BRCA2 mutation. The polygenic risk score is about 0.2% of the population. But at the same time, BRCA2 mutations are maybe, I don't want to give a number because I'm not on top of our BRCA2 epidemiology, but maybe not very far from that in terms of percentage of the population. So if you look prospectively, the predictive ability of PRS is not very far, and there is obviously much more from PRS to be given for people in the 5-10% quantile of the polygenic risk score. So my point is, I'm happy to take the discussion for the rare diseases, for the common traits. I mean, I think it's like for common cancers like breast cancer or colorectal cancer, it seems to me that the case is incredibly solid when you look at those data.
Wonderful. Okay, that makes a lot of sense. And thank you for explaining that. Right, we have 10 questions in the Q&A. So I think we'll try to do rapid fire question and answers if you can, Vincent. I think you could probably spend half an hour on each of these questions. The first one is from Isatu Sa. Actually, he's asked two questions. So polygenic risk scores may be used to estimate an individual's lifetime genetic risk of disease, but the current discriminative ability is low in the general population. Is there a corrective test for this? And also with rare variants, how do you differentiate them from a mutant of a transcript?
So I think the question is about the effect size of the PRS, and I think it refers, we're back to this issue of it's not that low. I mean, again, if you look at, at this high-level picture I gave you at the beginning, I think whether it's low or high it depends on the application and what you can do in terms of screening. And the point I'm making is that I think it's way enough to have substantial impact on screening and prevention strategy. In terms of the second question, how do you differentiate them from mutants of a transcript? All I've shown you right now is FH and BRCA. So for BRCA, it's fairly simple. We picked clear, well-annotated loss of function variants in BRCA2 for the graph I've shown you. And for FH, we've actually taken the list from the work of a paper published by Amit Kira and colleagues, who brought in experts in the FH field to diagnose those FH mutation. It's tough. It's a hard job to diagnose FH mutation. So we've taken advantage of that in that work. So we're not in the business of doing genetic diagnosis, but for the point of those talk, we've taken those shortcuts by taking very well annotated variants.
Okay, thank you, Vincent. That's a clear answer. Sorry if I get these names wrong. We have another two questions from Anupriya. So did you use array data and whole genome sequence data together in one analysis? And if yes, how did you compare the different QC metrics that exist in these two types of data?
So for analytical validation work, that's exactly what we do. We take sample, we use array data together with average data, and we see if there is concordance between the two. We do it for cell line, we do it for blood, we do it for saliva. It's part of all the regulatory layer you have to do to get the test, to be used in clinical trial in the community. So the answer is that that's what we've done. And there's a whole lot of stuff under the hood to make those things reasonable and work properly from a QC perspective.
Terrific. And also from the same person on kinship analysis, did you remove related individuals? To what extent did you remove different degrees of relatedness?
So it's straightforward to measure some sort of identity by descent or statistic across the population. Where did we exactly put the cutoff? I must say, I can't quite tell you from the top of my head. I suspect we went all the way to 3. That's probably as far as we've gone. But I just stop here and not say anything that might seem me of saying.
Sure. I mean, some of these questions coming up are very detailed. There's one from Helena about control cohort. So genomics is a disease cohort. What control cohorts did you use performing PRS with GeL data?
So GeL is it's a disease cohort for a lot of cancers, but it's also individuals who haven't had type 2 diabetes are controlled for individuals who have type 2 diabetes in the cohort. So basically, you can use the cohort as a whole and being smart about how you split your individual. So if you have cases and control built into the GeL environment. So we did not need to bring additional external control. It's all using non-case for a given disease to become control for this inside the GeL cohort.
Yeah, that's right. And we are introducing soon about 15,000 asymptomatic COVID patients as part of our COVID sequencing program. So that should contribute as useful control as well for people. From an anonymous attendee, the question is for cardiovascular disease. How many studies have reported minimal or no improvement in discrimination beyond the standard risk classifiers, such as Q risk? And this has little impact on interventions available. What is the value of PRS in cardiovascular disease? So it's a similar question to the first one, I think.
So I think there is indeed a fair and healthy debate around cardiovascular disease. I think part of the issue in those two papers, those two JAMA paper in particular, is the population you look at. So especially in young men, I think people who've looked at the data agree that the predictive impact is quite large. And in fact, almost a quarter of 40 to 55-year-old men who end up with a cardiovascular event with a coronary artery disease event in the next 10 years end up classified, meaning being pushed above risk threshold by the genetics. A quarter of those cases. I mean, if you look at the right slice of the population, the impact is actually quite substantial. I think an issue of the Mosley paper in particular is that they looked at fairly older population for which indeed the benefit of the PRS is less pronounced. But you ask me, where is it useful? The answer is in young men, it seems a substantial benefit in prediction in that population.
Has Genomics PLC introduced your own tools to perform PRS in GeL?
So yeah, so there's a whole piece about training, and obviously that's kind of what my team does, and we're doing that outside of GeL. But then we've imported our own computation tools, properly packaged and Dockerized and doing all the right things, their own environment inside the GeL, thanks to your team, Parker. So you're very helpful in helping us bring the right toolkit into the GeL research environment. And that's how we've done it indeed. Yeah, most of them have been optimized in-house for the work we want to do.
Okay, yes, you can containerize applications and bring them. Great. The next question, I think you've covered a fair amount, but do you have any other comments to make about the clinical use of PRS in the NHS?
I think I've commented on that quite a bit. I think it's case by case. I don't want to make a broad statement, PRS are ready or PRS are not ready. I think it's ready for specific use case. I think the heart trial, especially again in middle-aged adults, is a very reasonable and well supported by science thing to do. We can argue about other very specific use cases. I don't think we have the time to go one by one in that situation. No. But I do think there are many use cases that are ripe for clinical use today already. Indeed.
The second part of this question was really about data flows, and I expect what the questioner is getting at is: do you see this being implemented in primary care?
Yeah. So I think it's a primary care thing. I think it's exactly the way you go to the doc, you get your LDL levels, and you learn something about your risk. One day you go to your primary doctors, and you'll know about your genetic risk for CAD or whatever it is you want to look at, and we'll be dealing with it exactly in the same manner. So to us, it's mostly a primary care tool. And we think pretty soon we're going to be dealing with that exactly in the same way we did with other biomarkers. It is yet another biomarker, a good one you can measure early, but that's how we believe we should handle it.
Excellent. Thank you for handling these rapid fire questions. We've got more than usual and some quite difficult ones. Do you have any suggestions for incorporating rare variants into these models, given that common variants may modify penetrance of rare variants?
So predictive model combining rare and common variants is a fascinating piece of statistics. People who are interested should come work with us. It's great. So the problem is, until we had things like UK Biobank to really see in large prospective cohort how people are faring with those rare mutations, there is only so much we could do, and there are some good studies out there, but UK Biobank is really changing the game. So the answer is we need more prospective cohorts like UKB. I think is the only way to understand how those things work together. So we're working on it. We're working with other businesses to make that work effectively. Other cohorts, yeah, so we need more data. I think that is the main thing we're lacking to really nail those predictive analytics.
Excellent, thank you. I'm going to skip over a few of these just as we're running short on time. What is the best way to estimate the optimum sample size to derive PRS, especially in diseases with low prevalence, such as autoimmune disease? So this is about powering, something that we've been wondering internally as well.
To derive the PRS or to validate the PRS?
I think to derive the PRS. So to build a PRS, how do you power that type of calculation?
So I think to build the PRS, I'll take the same. I remember people asking me those questions the early day of GWAS. The answer is as many as you can. The more you have, the further you go. So I don't think I can give you a number. There are some very good papers out there showing, the sort of curve of performance given as a function of sample size. They keep increasing quite a long way into, when you have tens and tens of thousands of samples. So the answer is I'll pass and tell you as many as you can.
Yeah. Okay. Thank you. That would be actually be really helpful if you could post that paper in the chat when we get to the next speaker. Thank you. I think many people would love to read that. What is the percentage of non-coding variants you included in your scores?
So as far as PRS are concerned, we're completely oblivious to the fact that variants are coding or non-coding or synonymous or whatever they may be. They're essentially tagged for other variants. So the answer is that the vast majority of those variants are indeed non-coding. In a sense, we don't want to know. They tend to be common. So those models are ignoring that. Traditional PRS models are ignoring that feature of annotation.
Okay, and I think we will just use, we'll go for two more quick questions, and then we're going to move on to the next speaker. Thank you very much for this. So for cardiovascular disease, is the HEART trial enriched for young men? How much burden of disease will this capture? And is it offering screening to a small number of individuals? Or offering screening to a small number of individuals seems counterintuitive, question mark.
So I think, I don't want to say something wrong. I think the HEART trial is, I believe it's 40 to 60, but I'm not entirely sure. So I should double check my statement before giving you those numbers. So it's generally, on the slightly younger end of the spectrum, and depending on how you place it. And then it's up to the GP. I think the GP gets the results, and the GP inform his or her decision on the basis of the information. So for example, some individuals will be offered statin if they have high genetic risk for CAD or CVD. So that's the whole point of the HEART trial, is to see how the community uses this information and takes advantage of it. So more than more screening, it's more preventative actions in particular settings.
Terrific. And very last question, do you see a substantial differences in using ICD-10 codes as outcomes versus specifically built cohorts, i.e. where the complex disease diagnosis is confirmed by the clinician?
So ICD-10 codes are not perfect, and I do think that there is a drop in prediction for some traits where ICD-10 codes miss a little bit of the granularity of the disease. So I think, in a sense, all our estimates are a bit conservative. I think it's case by case. When you come to disease like breast cancer, people tend to know if they have breast cancer. It's not the sort of stuff you tend to not be aware of, so it's pretty reliable. We've seen examples of healthcare systems that are very poor at, for example, recording psychiatric diseases, schizophrenia, and bipolar disorder. In those cases, specific codes that have been cleaned up from a phenotypic point of view make a huge difference. So again, it varies a bit disease by disease. Some are easier to manage from an ICD-10 point of view. Some are very hard. So it depends a bit on what you're looking at.
Okay, Vincent, I think we will let you pause there for a breath. You've handled those questions really well. There were quite a few more, and perhaps we can send those to you by email to address. Quite a few comments also about the appropriateness of the use in clinic and whether these are validated tests yet. So I'm sure those will all be questions that not just Genomics PLC, but the NHS will be dealing with as we start to roll this out nationally. For now, though, just thank you very much for your time, Vincent. It was really fascinating to hear that. I'd love to introduce the next speakers, please.
Thank you.
Thank you. So Henry, Rebecca, and Daniel, terrific, you're all on the call. Thank you very much for joining us. So the second presentation is really a triple act with Professor Daniel Peckham, Dr. Rebecca Birch, and Dr. Henry Wood, who are jointly going to present on the link between colorectal cancer and cystic fibrosis. I won't introduce all three of you, but perhaps you could just give a little bit of your background when you start speaking so that we know where you're coming from. And over to you. We can see your slide. Excellent.
Well, thank you. Well, thank you very much for inviting us to come and give this talk. We're actually delighted to give a sort of summary of CFTR and colorectal cancer. And we said a missing link, which is really a question. Let's just, okay. I'm going to just quickly go an overview of cystic fibrosis. We're going to look at potentially the link between CFTR and cancer and also the background to the relationship. So what's really important to remember is actually cystic fibrosis in terms of the actual first mutation, the delta F508 mutation. It's relatively recently. It was first described in 1989 in this science paper. And what's extraordinary really is that we've got a condition that often people assume is very rare, but it is actually the most common autosomal recessive lethal condition in Caucasians. And 1 in 25 of the UK population actually carry a defective gene, a CFTR defective gene. And we now, since the 1989, we've got over 2,000 different mutations. The majority are F508 in the UK population, but there's a lot of differences. So for instance, if you take parts of breast or you go, for instance, to Israel, you'll see a very significant difference in terms of the types of mutations seen and the panels used to screen. Now, the important thing, of course, is that you'll all hear about basically cystic fibrosis causing mortality and morbidity through respiratory disease. So you get recurrent respiratory tract infections, progressive lung damage, and end-stage respiratory failure. Now, what is really important also to think about is the population. So the median age of the UK population is very young, it's 21, and that's very relevant and pertinent when it comes to talking about cancer. The median age of death is around 31, and the median predicted survival is around 50.6. And one of the key protectors in terms of infection within the lungs, of course, is our mucociliary clearance. We have, very importantly, airway surface liquid, which allows the cilia to beat normally. You've got the mucus, and we're constantly inhaling bacteria, fungi, and particles which stick to the mucus, and our escalator basically extracts it to the back of our throat where we swallow it. And this is a very important mechanism. And of course, in cystic fibrosis, it is defective. Okay, so the gene codes for CFTR, which is a cystic fibrosis transmembrane conductance regulator. It is a channel. And if we look at the lungs, it is within the, for instance, airway epithelial cells. It is very important as acting as a chloride channel, and it also regulates bicarb and has a very important inhibitory effect on the ENaC channel, the sodium, the epithelial sodium channel, and therefore thereby regulates hydration and the airway surface liquid. So if you've got a whole lot of cells, for instance, with cystic fibrosis and the healthy cells, and you poured liquid on it, you'd find you get dehydrated airway surface liquid in the CF cells. Now, the most common mutation, the F508, the actual protein produced is so abnormal that it actually doesn't get really to the apical membrane. So therefore, what happens is you actually have an absence of chloride secretion, you have defective acidity, and you also take the brakes off the sodium. And that sodium flux is extremely important in drawing water and also developing airways dehydration. Now, arbitrarily, we tend to classify mutations as clinicians into 7 classes. And the class 1 is the classical sort of premature stop codons, where you don't actually get the production of protein and actually is associated with very severe CF disease. Class 2 is the delta F508, and that's associated with processing. But what's really important is this is arbitrary. I say arbitrary because things like delta F508 will also have dysfunction. So it's got class 4, etc. Class 3 is the classical gating mutations, going all the way to basically class 7 mutations. Now, the amazing thing, I suppose, is that in 1989, we discovered the first mutation, but in 2011, we discovered the first key treatment that could actually radically improve cystic fibrosis. And this is a potentiator, a small molecule, which basically allows, if you can imagine that in a class 3 mutation, it's like a gate, it doesn't let the cow out or doesn't let the ions out. And what happens is that you get significant severe cystic fibrosis features. But they found that the addition of a tablet called ivacaftor, it increased CFTR expression, increased ion transport, had a dramatic improvement in lung function of up to 10%, and a significant reduction in sweat tests. The problem is, of course, is if you use a potentiator in delta F508, you haven't actually got the protein there to stimulate. So more recently, very excitingly, and I'm sure a lot of you will have read it in the journals, that a triple therapy has been introduced clinically into the UK and parts of Europe and America. And this is using tezacaftor and lexicaftor, 2 other small molecules, to stabilize the protein, allow this protein to be expressed. And then what we use is a potentiator to stimulate that protein. And this, again, has been shown to have a significant impact where you can see, even in a heterozygote patient, a dramatic improvement in lung function, FEV1, and quite a marked reduction in sweat chloride. And that's really important when it comes to what we're talking about bowels, is that CFTR, the small molecules, the CFTR modulators, act within the majority of cells within the body. So the hypothesis I've given you is a classic one we say in terms of what goes on in cystic fibrosis, all about airway surface dehydration, pH and reduction in defensins, etc. But it's a lot more complicated, as you expect. CFTR itself is probably involved with autophagy, which is probably defective. It is associated with increased inflammation and ER stress. It is associated with abnormality in a whole series of other intracellular mechanisms. So cystic fibrosis is not just about the lungs, it is a multi-system disease because the CFTR is everywhere. So a classic example, just as from a clinician's perspective, is all our patients on Kaftrio, female patients, getting pregnant. And they're getting pregnant because actually it's changing the cervical secretions. So you can see that really these CFTR modulators are impacting throughout the body. But of course, importantly, it is CFTRs in the intestine. This is just radiologically showing this is dilatation of the airways, which is called bronchiectasis for non-clinicians. And here you can see these dilated airways, which is a classical feature you get in the lungs of cystic fibrosis. Now, we often spend 40% of our time on the GI system. And this is important again, because individuals with cystic fibrosis get a lot of symptoms. We're doing presently a large study on the gut microbiome, looking at symptomology and diet, and it's quite noticeable how frequent these symptoms are. And one of the complications due to the thickened secretions and abnormal ion transport and abnormal mucus within the intestines is you get a condition called distal intestinal obstruction syndrome, where you can see you get this intense fecal loading, which occurs at the terminal ileum, cecum, and then large gut. And this can cause acute bowel obstructions, which is important. And of course, a lot of these symptoms are the features. Sometimes you get changes in bowel habit, which would be the features you might think about if somebody developed a sudden change when they'd got malignancy. Now, one of the, I suppose, heroes of cystic fibrosis was in the 70s was a doctor called Dr. Crozier, who basically really introduced high-fat diets with pancreatic enzymes. And by really introducing it, he changed the body composition quite significantly and improved clinical outcome. And so over the decades, we basically improved the survival of individuals with conventional therapy dramatically, you know, from really a few years to predicted life expectancy in 50s. But we've done this through also diet. And of course, this is the classical diet, very little fiber, very high fat, high carbs, and high protein. And of course, this is a very bad diet when it comes to thinking about colorectal cancer. So the pressures also of antibiotics, PPIs, diet, host genetics, you know, pancreatic insufficiency leads to a pro-inflammatory gut microbiome. And as we know, gut microbiome is associated with many diseases, including colorectal cancer. Not only that, in cystic fibrosis is really has a sort of auto-inflammatory element. It is associated with inflammation in lots of tissues, including the gut. And one of the actual pathways which is hyperactivated is NLRP3 inflammasome, whereby LPS, for instance, from the gut bacteria or lung gut bacteria can trigger off a hyper responsiveness in terms of activation with high productions IL-1 beta, IL-18, caspase 1. And if you look at this, this is individuals who starting on Simkevi, which was the original dual therapy, but this is exactly the same for Kaftrio. And you can see that if you look at the blood sample levels of IL-18, IL-1 beta, you can see that levels are going down. And some of the baseline levels are much more similar to sort of SEDS, systemic autoinflammatory diseases. So there's a component there. And if you get monocytes or airway epithelial cells and you try and stimulate them with LPS, what you find is you get a hyperresponsiveness. But once you start adding in the Simkevi, the drug, the CFTR modulators, you find by 3 months you've got a massive reduction in terms of the inflammatory response. And I think this is very important because inflammation is probably important within possible risk factors for colorectal cancer. So what we've got here in terms of gut inflammation, we know, and we've known for years, that fecal calprotectin is very high. We know that there is a pro-inflammatory gut microbiota. We know that there's a low fiber and there's a dysfunction, probably short-chain fatty acids. We know there's inflammasome activation, and actually there's an increased prevalence of Crohn's disease. And so I've drawn a little sort of picture just to sort of demonstrate what's really going on. So we can just say, okay, fine, in cystic fibrosis, the problem is that we've actually got these influences. We've got the pancreatic insufficiency, the antibiotics, the proton pump inhibitors, diets, 50% have diabetes. We've got inflammation, dysbiosis, reduced short-chain fatty acids, all of which could potentially increase the risk of colorectal cancer. But what we mustn't forget is at the cellular level, CFTR is very important because CFTR is driving inflammation. There's a hyperactivation inflammation throughout the body. There's also dehydration. So CFTR, chloride bicarb, so you can imagine the gut's not protected, and also the mucin. Mucin is secreted via CFTR calcium-mediated effect. And if you actually look at some of the videos of it being produced, it just sort of flops out and doesn't actually get to the right place. So what we've got is a barrier, the barrier has been damaged. We then also have CFTR bicarb, very important in tight junctions. They're also damaged. Then we also know that CFTR could be a tumor suppressor gene. And interesting enough, colorectal cancer downregulates CFTR, and this may be associated with worse prognosis, worse outcome. And then, of course, we've got activation of a whole host of things, ROS, NLRP3. And then, of course, we've got reduction in transit times because CFTR is actually within the mesenteric ganglia. So, you know, if you ask a clinician such as myself who's been doing cystic fibrosis for well over 25 years, you know, for me, seeing cancer in CF is not unusual, but seeing it in bronchiectasis, PCD, etc., is incredibly unusual. And this is a paper that was 2007, so looking back 10 years from 2007, and we had, as you can see here, 3 cases were diagnosed with malignant condition without solid organ transplants. 1 case was a pancreatic carcinoma in an 18-year-old. We had esophageal carcinoma, and we also had a bowel cancer in these early days when people were very, very young, and the population would have probably been more pediatrics than adults. Then looking at this in more detail, looking at the CF registry, which is one of the most in-depth registries around, cancer really wasn't really logged in terms of conditions. So for instance, it's been poorly entered. We don't have a specific entry for around between 40 and 50% of cases, precisely what the primary cancer was. But certainly in this, there was a increased risk of colorectal cancer. Then one of the most important studies was this study from looking at the American CF Foundation registry. And again, there was elevated risk of digestive cancer. A further study by the same first author looked at it in both non-transplant and transplant patients and found that there's a high expected incidence of cancer of the digestive tract in both cohorts. And importantly, in this paper, it shows that if you look at just transplant patients, CF individuals with transplants have a high incidence of colorectal cancer versus other people who have had a lung transplant. So CF itself seems to have a predisposition to colorectal cancer. So these studies were done in the USA, and really there isn't any linked cancer registry data so far in England. So I'm going to pass over now to my colleague.
So I'm a big data scientist, so slightly different background. And what we set out to do with this piece of work was try and address the gap in the knowledge around the cystic fibrosis population in England. So we wanted to quantify any excess risk of colorectal cancer and then look at the associations between mutations of the CFTR gene and colorectal cancer, particularly in carriers. Next slide, please. So we used CorrectR to look at the cancer population. This is an England-wide data repository, and it includes linked HES and cancer registration information. This is important because it's the gold standard for cancer registration information that's held, including things like site of tumour, stage of disease, and how patients were diagnosed. It includes all individuals who were diagnosed with a colorectal cancer or CRC in the English NHS. Next slide, please. And what we've done is we've used this data, so we have details about the date of cancer diagnosis. In order to define somebody as having cystic fibrosis, we don't have the date they were diagnosed with that. What we've done is look for a record of cystic fibrosis that's been reported during admission to hospital in the period prior to the cancer diagnosis. So it's important to note that we know if someone's had cystic fibrosis, we don't know the duration they've had it for. Next slide, please. So what we did with this cancer data is we looked at all patients who were diagnosed with a colorectal cancer between 2007 and 2017 in England, who were diagnosed within the English NHS. Individuals who had fewer than 2 records of cystic fibrosis in the period prior to their cancer diagnosis were excluded from the sample. This is because there's a study that suggested that all individuals with cystic fibrosis are likely to be admitted to hospital at least once a year. And also when we looked at the population who only had 1 record, their median age was a lot higher than we'd expect for the cystic fibrosis population. So to ensure that we've got the group who we thought we were looking at, we've restricted it to only those with 2 or more records. We calculated a crude instance of colorectal cancer per 1,000 individuals, and we chose 1,000 because of the relatively small size of the CF population in England. We validated this crude instance rate using the UK Cystic Fibrosis Registry dataset, who are the gold standard for cystic fibrosis records. So between the two, if we get the same answer, we've got confidence in our results. We age standardised the rates to the European standard population because we're aware that there's a very different age distribution in the colorectal cancer population compared to the cystic fibrosis population, as Daniel said. There were fewer than 5 cases of colorectal cancer in those with cystic fibrosis who'd had a solid organ transplant, so there wasn't enough power in this for statistical testing. So you'll see that I don't mention that in the results from this part of the study. Those patients were included just as in the whole population. Next slide, please. So the characteristics of what we found. We got 28 cases of colorectal cancer with a record of cystic fibrosis compared to over 350,000 who didn't. Those with cystic fibrosis, we had a higher proportion of females. So we had 60.7% who were female compared to 44.5% in the population who didn't have CF, and a higher proportion of right-sided tumours. So we had 50% in the CF population versus 26% in those without CF. And that's important because that is thought to be related not only to the microbiome, but also the inflammation response in the patients. Next slide, please. When we just looked at crude incidence rate across the whole population, the crude incidence rate of CRC was higher in those without CF. Because of the significant difference between the age populations, we thought this was likely to be led by the age profile of the population. So those without CF were likely to be older, therefore more chance of CRC generally. Next slide, please. When we split it by age group, we actually found that there were 11 additional cases for every 1,000 individuals aged over 50 in the cystic fibrosis group. So you can see that the age distribution here is having a significant impact. Next slide, please. When we standardised the incidence to the European population, there was actually a 7-fold increased risk of colorectal cancer in those with cystic fibrosis. So we got 53.9 per 1,000 individuals in the CF population compared to 7.37 in those without CF. Next slide, please. And then I'm moving over to Henry now, who's going to present the Genomics England section of the analysis.
Thanks, Becky. So my part of the work is looking, as Becky said, at the Genomics England data. And this isn't looking at people who have cystic fibrosis, it's specifically looking at people who carry the gene. So my part of this is I'm part of the 100,000 genome colorectal cancer cohort analysis team. And so part of that analysis, we're basically looking at what causes cancer in these people. So we're looking at mutations in cells in the gut that are giving rise to people's tumors. And for those of you that aren't familiar with cancer genomics, you tend to uncover thousands and thousands of variants per person. And these are mostly just germline things that people are born with. So to distinguish the somatic things that are causing the cancer and the germline things that people are born with, you have to sequence the blood samples as well. And generally, that's where people leave the blood samples. So there's a vast data source that's not generally used for much. So what we thought was this represents a really valuable untapped resource. So we've decided to repurpose it. It's essentially free data. It's sitting there on a disk. So we're using it to look at genetic differences between the people who've got colorectal cancer and the people that don't. And specifically in this project, do these people have an increased instance of carrying variants associated with cystic fibrosis? So next slide, please. So in terms of methodology, I just focused on the CFTR gene in the 2023 colorectal cancer patients in this cohort, looking at their blood samples. And I just did a simple filtering of 10% variant allele frequency to get rid of noise and errors. And I matched all these variants to known variants in the CFTR2 database, which is a database of all known CFTR variants, and it classifies things as CF causing, non-CF causing, mixed effect, that sort of thing. So I got rid of all the variants that we knew weren't associated with cystic fibrosis. So next slide, please. So this is the top of our results table. And what I simply did was I counted all the variants and counted how many patients each variant was found in. So you can see that nearly 1,900 people were wild type for the cystic fibrosis gene. So they had no variant, no mutation at all. 141 people had something, so some sort of variant. And then I just go down the list of the most common gene was found in 62 people, the second most common variant in 18 people, and so on until we found that at the bottom of the table, there's 24 mutations were found in one patient each. And I just want to focus on 2 lines of this. So next slide, please. So the most common variant in cystic fibrosis, or causes cystic fibrosis, is the F508 deletion that Daniel was talking about. This is present at 3.1% in the general population, and it's present at 3.1% in our cohort, so exactly the same. So next slide, please. But if we look at all heterozygous mutations in the cystic fibrosis gene, they're present at 4% in the general population and 6.9% in the Genomics England colorectal cancer cohort, so much, much higher. So next slide, please. So just to summarize the Genomics England part of the story, we found that the most common F508 deletion variant has the same frequency in colorectal cancer patients as the general population, but that the total numbers of potentially damaging variants in this gene is 70% more common in colorectal cancer patients than you'd expect. And with that, I'll hand back to Daniel and Becky for the summary.
It's misbehaving, I'm sorry. So I suppose the key conclusions are that the incidence of colorectal cancer was significantly higher among those with CF when compared to those without. That it has seemed to occur in younger individuals. There was a predominance of right-sided tumor, which is relevant because CFTR expression is higher as you go from the right to the left, and also potentially inflammation. And the prevalence of CFTR mutations are higher than expected among individuals with colorectal cancer, although it's very noticeable as the R117H, and we would have expected a higher prevalence of the G551D. So what are the implications? I think that there are significant implications because, firstly, what is the impact of CFTR on the natural history of colorectal cancer? We talked about cancer, but in terms of if you actually have a mutation, you reduce the expression of CFTR, actually is the outcome different within individuals with those mutations? In terms of, is it a biomarker potentially for early screening for individuals? Could low-dose CFTR modulators actually reduce the risk? We know that, for instance, the actual tumors downregulate CFTR, so is there actually a role? And what is actually the role of CFTR, actually, if it is at all, in terms of causing or being related to cancer? And does it actually tell us some of the etiology? And is it possible that we could, as again, could we actually reduce the risk in terms of low-dose modulators, or actually use CFTR modulators potentially to improve outcome clinically? So those are the acknowledgements, and we'd like to thank you very much for listening to this talk, and we're open to questions.
Well, thank you very much, all 3 of you, for a very interesting talk and what seems to be a very good use for the Genomics England dataset. Fascinating, the link between those 2 indications. We've had only really 1 question on the Q&A, so can I just encourage people to ask questions? I will come to that question. Also, our Chief Scientist, Matt Brown, is on the line. I know he has a few that he'll ask over the chat. Maybe a question from me, which was provoked just by your slide right at the end, where you were talking about biomarkers for potential screening. For any of our participants that are listening to this, what would you say to encourage them about the prospects for cystic fibrosis sufferers, and how have you engaged with patients to make this real for someone on the CF pathway?
Is that to me?
Any of you could.
Okay, so I think, okay, so what's really important is that for the last 10 years, I've been trying to get gastroenterologists to do colonoscopies on my patients. And really, everybody reads the articles from America and say it's not true, they don't believe it. And so it's taken a long, long time for us to actually find out. For me, I think the problem is we've got to take, it's understanding the sort of the statistics, epidemiology. We've got a population with the average age of 21. And we've also, the exciting thing is we've got these new therapies. And the real problem is these CFTR therapies are likely to increase life expectancy by 30 years, 40 years potentially. So we're going to massively increase our population. And it could be that CFTR modulators reduce the prevalence and incidence of bowel cancer. But unfortunately, the increase in age is probably going to outweigh any benefit. So somebody could, for instance, in the future say, well, they started CFTR modulators and have an increased risk of cancer. So therefore, CFTR modulators cause cancer. And so I think what we really do need to do is understand the epidemiology, be able to predict what we think will happen with time, and we can use these models. We're going to go into a totally different perspective status to actually really start exploring this in much more detail. That's what we want to do. And so I think that so at the moment we are doing colonoscopies. The average is from 40 years is what the Americans suggest, 30 years for transplant, because of course the transplant patients are very high risk. And we are starting to see, we are starting to pick up early malignancies. So I think, unfortunately, we've got to watch the natural history. But what's happened at the moment that's so important is life expectancy is going to be dramatically increased. And therefore, this becomes an even more important problem.
Okay, that's really clear. So in fact, this data really does strengthen the evidence for the use of colonoscopies in kind of in patients as they're progressing and living with CF. Thank you very much for that. Can I move on to a question by Daniel Peckham, who's one of the heavy users of our data? He's asked, have you checked other populations in GeL to see if the increase in frequency in CRC is specific? And actually that goes quite well with another question by Uma, where she's asked, there's an increasing interest with the incidence of cancers and some lysosomal disorders. My understanding is that lysosomal acidification is impaired with the CFTR variants. Could lysosomal dysfunction be contributing to this increased cancer risk?
So I can take Dan's side of things. We haven't checked in other cancers just for technical reasons that I'm only a member of the colorectal cancer GSIP, so I don't have access to all the other the files. So maybe I could talk to Dan offline and we could arrange, he could check the other cancers for me.
Yeah, and Henry, we can certainly organise that for you at Genomics England. The purpose is that you should be able to do pan-cancer research.
Yeah.
And to the second part of the question by Uma about lysosomal dysfunction contributing to an increased cancer risk.
Yeah, I think absolutely. I think that we're just beginning to really understand CFTR dysfunction. So for instance, CFTR in terms of lysosomes, there's probably abnormality there. In fact, one of my PhD students doing some work on that. There's dysfunction in mitochondria. There's a whole host of things. And I think absolutely this is something we need to do. So I think what's really fascinating about the early data is firstly, we want to look at all the cancers because there's lots of other cancers that is probably associated with CFTR. And it could be that CFTR mutations actually protect some cancers. But it's really confusing. So a good example, we see skin cancer, but the problem is we use a lot of voriconazole, UV hypersensitivity, transplantations, lymphomas. So but things like cervical cancer, breast cancer potentially might be important. But I think this understanding at a cellular level is really, really important. And that's why I think it's so exciting. If there really is an association, for instance, colorectal cancer CFTR, the question is, why is it? It's not likely just to be due to airway surface liquid. It's probably to do with lysosomal dysfunction, mitochondrial dysfunction, inflammation, a whole host of other things. And that's what we need to go into the labs for and try and find out what it is. So if somebody would like to come and work with us, we'd love to do some collaboration.
Okay, excellent. Thank you. And I think this is more of a comment by Syed, but I'm pleased to clarify. Syed mentions that gain lift charts may be of more practical value than ROC or AUC curves. I'm not sure if that makes sense to you guys.
May have been a comment for Vincent.
Oh, no, that just came through now. Matt, so let me introduce Matt Brown, our Chief Scientist, onto the line. I can see your hand is up. Please go ahead, Matt. Yeah,
hi, thanks. Fascinating presentation. So look, I was really interested by the suggestion that maybe CFTR might have a relevance to colorectal cancer in non-CF patients. So the heterozygotes have an increased risk, and the possibility that CF treatments might therefore be have some benefit in preventing colorectal cancer in perhaps that subset of the population. I'm wondering, do the colorectal cancer GWASs that have been done to date show any signal around CFTR?
Well, in terms of function, I mean, the thing I would say is if we look at the mutations, we've got the Delta F508, which is the classic. But then the next one that came, which is really intriguing, and I can't explain why, is R117H. Now, the R117H is very interesting because that's linked to the polythymidine sequence. So if you have a 5T R117H, it's much more significant because it's partially functional. So the amount of R117H will be dependent on the polythymidine sequence. We couldn't get that data. We couldn't get the polythymidine sequence. I'm not sure why. We would expect the things like G5. So there are certain mutations that are missing that I can't explain why. So it's a very complicated area. So
So my question was actually to do with GWAS data in colorectal cancer and whether it picked up any CFTR signal, because that would support the hypothesis of what Henry's working on.
I don't know, Henry.
Yeah.
I've not looked at GWAS data at all, really.
Okay, well, that might be something worth doing. I'm also just interested in coming back about the mechanism that you're proposing about how this might actually lead to, how the CFTR variants might lead to colorectal cancer increased risk. And I'm wondering if you could expand a bit on that.
Yeah, so I suppose the question we have to ask ourselves is, why do 1 in 25 of us carry this gene? There must be advantages and there must be disadvantages. And, you know, if you ask me why, I mean, I have a mad hypothesis, and I'll tell you my hypothesis, is that actually maybe, well, firstly, they think that it could potentially be things like cholera and typhoid. So actually the gut again, in terms of hydration and not getting severely dehydrated during infections. But I do wonder whether, we know that carriers have increased frequency of lots of things. They have increased risk of bronchiectasis, increased risk of COPD. But is it possible that carriers actually have a hyperinflammatory response that can be actually protective? And so we don't know. I mean, for instance, I'd love to, I mean, I've asked a question in COVID, but I haven't got an answer, is what is the prevalence of CFTR carriers in COVID, the ones who die and survive? Now the data, you know, and it's so hard getting this sort of information out of people. There is some data suggesting that carriers are worse in Italy and some that says it's better. But, you know, when we had, for instance, the flu in the First World War, was that protective? So we don't actually quite understand why there's a protection, and that's the key thing. But it's so common. It could be that we've accidentally found it, but I think that others are beginning to show the same thing. So
right,
yeah. And in terms of
so do you have an explanation for the north-south gradient that you see with CFTR variants? Which is, I guess, it's a similar question. Is, you know, is there, what's driving the high population prevalence of CFTR variants, and in turn, why do we see population gradients in it?
Yeah, I mean, is it possible, for instance, that the carriers of CFTR, so a carrier of CFTR will have about 50% CFTR function. So is it possible that actually if you have reduced CFTR function, you have increased inflammation? And that very subtlety superimposed with other risk factors is potentially, so it's not necessarily that CFTR carrier status causes colorectal cancer. It might accentuate other things. And that could certainly be a process, but also we know that colorectal cancer downregulates the CFTR gene. So is it possible that if you've actually got reduced CFTR and you get, for instance, early changes, that maybe accentuates its development? So these are total hypotheses. Nobody knows. People are trying to work on it. There's a Cambridge group working on it with Sheffield at the moment.
Daniel, I would have thought our COVID data, which is a cohort of asymptomatic mild patients and then an acutely ill cohort, would be quite good at looking at the resilience conferred by the CFTR gene potentially.
Well, that'd be wonderful if we could do that. That'd be really exciting.
Talk to our team. We can certainly support with that. That would be great.
That'd be great.
I've got a question from Annalise, which is to do with carrier status. Annalise, why don't you just ask that question out loud? Feel free to unmute yourself. Annalise is part of our rare disease team at Genomics England. Okay, if you can't unmute yourself, I'll read it out. So there is some evidence that.
Sorry, I've just been granted access to my to unmute. Thanks, Parker. It was just that I'm really interested as a sort of clinical geneticist in the sort of carrier status and how that in itself has been linked to lots of other gastric, respiratory, etc. excess when people look at large numbers of electronic health records. And I just wondered if this was also the case, if you thought this might also be the case for other cancers, that carriers might also have a higher incidence.
Absolutely. Our plan is absolutely to look into this in much more detail and not in depth. So what we plan to do is link into the CF registry. So we can actually identify every single individual CF for the last decades. And that's the way we need to do it, link the registries. And by doing that, we can then identify which are associated with CF. And then we've got ideas in terms of looking at carrier status in lots of different cancers. I think we need to do this in a prospective, sensible way. I think this is the time to do it, especially with the fact that we've got these new modulators that we can actually do something about it. But yeah, it's fascinating, isn't it? I mean, we're seeing now modulators being used, for instance, in COPD, trials in bronchiectasis. We know that smoking reduces your CFTR function, fascinating in lung cancer. So there's so many fascinating things, but again, this is all hypothesis and ideas and concepts. Does that answer your question, Anna?
Yes, thank you very much. I do think it's fascinating. Thank you.
Okay, well, I think unless there are any other questions, and please do put your hands up or just ask in the chat. I think we've come to the end of that this seminar. That was a fascinating talk. Thank you to all three of you, Daniel, Rebecca, and Samuel, and also thank you, Vincent. I think it was great that there was not only a link between colorectal cancer and cystic fibrosis here, but I mean, very interestingly, Genomics PLC work closely with Vertex, who of course are large in the cystic fibrosis space, and I mean, just having the two of you on the line by coincidence, that was not done by design, you know, I think suggests lots of opportunity for a clinical academic collaboration, which is something we so greatly want to happen within our communities of academic and biopharma users. So wonderful to have you both on the line with such clear presentations. So thank you very much to all of the speakers. Can I just remind our audience of next month's talk? So I mentioned at the beginning, but we have a teach-in on the use of our data set on the 15th of March. If you go back to the seminar slide, Zan, Professor Bill Newman talking about mitochondrial dysfunction as well. And can I also do a special call out for our research summits? So I mentioned it before, we are doing a call for abstracts. We've actually extended the deadline for that call. It closes this Friday. So please do email any abstracts for people that want to present to events@genomicsengland.co.uk. Registration is open. We've got some very, very strong candidates already. There'll be a poster hall for submitting posters as well, but we want to have some of our top researchers on the stage presenting to about 1,000 people in person. We expect multiples of that on the line. I hope you will go and check out our YouTube channel where you can see the past Genomics England seminars. They're fascinating, and please do join us for the next one. And with that, final thank you to my team at Genomics England for organizing this for the speakers today, and I'll close out the call. Thank you very much, everyone.